DNA is the abbreviation of deoxyribonucleic acid. DNA stores the genetic information that is responsible for the growth, development and functioning of the living system. DNA is transferred into RNA and the genetic information is passed by this transformation. DNA generally exists as a double stranded structure. Both strands of the DNA molecule always coil together to form a double helix. Each strand of the DNA molecule is formed by a chain of 4 nucleotides. A nucleobase, a phosphate and a deoxyribose are present in DNA's nucleotides. The 4 nucleotides present 4 nucleobases. They are adenine, guanine, thymine and cytosine. The nucleobases are generally abbreviated as A, G, T and C. Purine bases are adenine and guanine, whereas pyrimidines are cytosine and thymine. Phosphodiester bonds are formed by these nucleotides. These bonds create a phosphate deoxyribose, which is the backbone of the DNA helix. Through hydrogen bonds, base pairs are formed. Adenine always pairs with thymine and cytosine always pairs with guanine. DNA strands have different ends of a single strand. They are 3' and 5' end. By adding nucleotides to 3' end of the DNA strand, DNA polymerase can synthesize DNA in only one direction. The pairing through hydrogen bonding shows that the information in each strand is surplus. The flow of genetic information within the cell is stated as the central dogma of life. In the year 1958, the central dogma of life was stated by Francis Crick. The central dogma of life can also be described as DNA makes RNA, RNA makes protein. The general pathway of information through replication, transcription and translation is shown by the central dogma. The process of developing two same exact or identical copies of DNA molecule from one original DNA molecule is known as DNA replication. The process is the basis of biological inheritance and it occurs and happens in all the living organisms. Biological inheritance is passing the traits to its offspring by their ancestor or parents. As we already know, DNA consists of 2 strands. Each strand of the original DNA acts as a template which is used in the production of the complementary strand. This process can be referred as semi-conservative replication. The mechanisms like error checking and cellular proofreading assure the accuracy for DNA replication. At a specific location, DNA replication starts in a cell. In a genome, from a specific sequence, the replication is initiated. This is called as origin of replication. Relaxation of the DNA molecule in the origin and production of new strands results in the formation of a structure within the nucleus. The structure formed may grow bidirectionally from the origin. This kind of structure formation within a nucleus is known as replication fork. A number of proteins are related with the replication fork which helps in the initiation and continuation of the DNA synthesis. By adding complementary bases to the template strand, DNA polymerase synthesizes the new DNA molecule. DNA replication can be performed artificially outside the living organism. In a template DNA molecule, the DNA synthesis at known sequences can be initiated by using artificial DNA primers and by isolating the DNA polymerases from cells. The polymerase chain reaction uses the artificial synthesis techniques to work on a particular DNA from a pool of DNA molecules. DNA polymerases are family of enzymes that go through all forms of DNA replications. DNA polymerases cannot initiate the production of new strands. They can extend only an existing DNA or RNA strand that is paired with a template strand. To initiate the production, a very small fragment of RNA must be created and paired with the template DNA strand. The small fragments of RNA are called as primers. By extending the 3' end of the existing nucleotide chain, DNA polymerase synthesizes a new strand of DNA. This is done by adding new nucleotides that match the template strand through the creation of phosphodiester bond. For this process of DNA polymerization, the energy comes from the hydrolysis of high energy phosphate bonds. The high energy phosphate bonds lie between 3 phosphate groups that are attached to unincorporated base. To a growing DNA strand, when a nucleotide is added, the formation of the phosphodiester bond between the phosphate group and the growing DNA chain is supplemented by a high energy phosphate bond. This will further release the 2 distal phosphates as a pyrophosphate. Mostly, DNA polymerase is highly accurate. For every 107 nucleotides added, it has an intrinsic error rate of less than 1 mistake. Some DNA polymerases reactions also have the ability of proofreading. In order to correct the mismatched bases, they can remove the nucleotides from the growing strand. Post replication mismatch repair mechanisms finally monitor the errors in the DNA molecules. This has the capability of identifying mismatches from the newly synthesized DNA strand. The rate of DNA replication was first measured in phage infected E. coli as the rate of phage T4 DNA elongation. During DNA replication, a replication fork structure is formed within the nucleus. This structure is created by helicases. Helicases breaks the hydrogen bonds that hold the 2 DNA strands together. The structure formed has 2 branching prongs. Each prong is made up of strand of DNA. For leading strands and lagging strands, these 2 strands functions as a template. The templates must be properly referred to as leading strand template and lagging strand template. In the 5' to 3' direction, DNA is synthesized. At the replication fork, the lagging strand templates and leading strand templates are oriented in the opposite directions. The major issue is to achieve the production of the new lagging DNA strand. The growing replication fork's production is opposite to the production of the new lagging DNA strand. The strand of the new DNA that is being produced in the same direction as the growing replication fork is known as the leading strand. The leading strand template is read by a polymerase and complementary nucleotides are added to new leading strand on the continuous basis. In prokaryotes, DNA polymerase 3 is the polymerase that involves in the production of leading strand, and in yeast polymerase epsilon is the polymerase that involves in the production of leading strand. In humans, within the nucleus, pole epsilon and pole delta are involved in the production of leading strands and lagging strands, where in the mitochondria pole gamma is involved. In some circumstances, pole epsilon can be substituted for pole delta. The strand of the new DNA that is being produced in the opposite direction as the growing replication fork is known as the lagging strand. The replication of the lagging strand is more complicated than the leading strand because of its orientation. The production of the lagging strand is short and separated segments. The template DNA is read by the primase on the lagging strand. This initiates the synthesis of the short complementary RNA primer. The primed segments are extended by a DNA polymerase, which forms Okazaki fragments. The RNA primers are removed. The RNA primers are replaced with DNA. DNA ligase is used to join the fragments of DNA. The DNA replication process undergoes 3 coordinated steps. The steps are initiation, elongation and termination. A cell must replicate its DNA first in order to divide. In a DNA molecule, this process is started at specific points. This is known as origins. The origins are targeted by initiator proteins. In yeast, this protein is origin recognition complex, and in E. coli, this protein is DNA. Initiator proteins use the sequences that are tending to be AT rich. Because 2 hydrogen bonds are present in AT base pairs, which are easier to unarchive. To unzip the double stranded DNA, the initiator proteins form the pre-replication complex by recruiting other proteins. This is done once the origin has been situated. Before the synthesis is initiated, a free 3-hydroxyl group is required by all known DNA replication system. DNA is always read in 3' to 5' where a new strand is produced from 5' to 3' direction. 4 clearly outlined mechanisms have been described for synthesis. All cellular life forms use a primase to produce a short RNA primer. Their synthesis occurs within a free 3'OH group, which is later on elongated by a DNA polymerase. The retrosec elements utilize a transfer RNA that grounds DNA replication by supplying a free 3'OH. In the adenovirus of the bacteriophages, an amino acids side chain provides the 3'OH group to which nucleotides are added to form a new DNA strand. The nucleotides are added by the DNA polymerases. The rolling circle replication, shortly called as RCR, endonuclease creates a small cut in the genome strand or on the DNA molecule strand. This is the mechanism of rolling circle replication. On the nuclease, the nicked strands 5' end is transferred to a tyrosine residue. The DNA polymerases then uses the 3'OH group for new strand synthesis. The cellular organisms use the first mechanism because it is the well known best mechanism of all the 4. The two strands are first separated in this mechanism. To the template strand, RNA primers are added by the primase. One RNA primer is received by the leading strand and several RNA primers are received by the lagging strand. By a high processivity using replicative DNA polymerase, the leading strand is extended from the primer continuously. This is the replicative polymerase. From each primer, the lagging strand is extended discontinuously, thus forming Okazaki fragments. Primer RNA fragments removes the RNAs. A low processivity DNA polymerase enters and fills the gaps. This DNA polymerase is distinct from the replicative polymerase. When the above process is complete, on the leading strand we can find a single nick and on the lagging strand several nicks. The newly replicated DNA molecule is completed by filling the nicks. The nicks are filled by the enzyme ligase. Between bacteria, archaea and eukaryotes, the primase used in this process varies significantly. The primase used by bacteria belongs to the DNA G protein superfamily. The DNA G protein comprises of a catalytic domain of the top rim fold type. The primase used in this process by archaea and eukaryotes is a highly derived version of RNA recognition motif, that is RRM. To many RNA polymerases, RNA dependent RNA polymerases, this primase is also structurally similar to many reverse transcriptases, DNA polymerases and cyclic nucleotide yielding cyclases. A nucleotide transfer mechanism is shared by all the protein above. In the DNA replication process, multiple roles are taken by multiple DNA polymerases. The polymerase enzyme DNA pol 3 in E. coli is responsible for DNA replication. At the replication fork, it assembles into a replication complex. This demonstrates extremely highly processivity and remains intact for the total replication cycle. The enzyme DNA pol 1 is responsible for substituting RNA primers with DNA. In addition to its polymerase activity, DNA pol 1 has 5' to 3' exonuclease activity. Using the exonuclease activity, it also extends the DNA strand behind and degrades the RNA primers ahead of it. The process is called as nick translation. Then pol 3 pol 1 is very much less processive, because the primary function of pol 1 enzyme is to create short DNA regions rather than creating very long DNA regions. Pol alpha, the low processivity initiating enzyme, has intrinsic primase activity in eukaryotes. Pol delta and pol epsilon are high processivity extension enzymes. During the DNA synthesis, the DNA original strands continue to relax or unwind of the bubble on each side. This forms the replication forks with two projections or prongs. On the circular chromosome, bacteria have a single origin of replication. A theta structure is created by this process. Replication is created at multiple regions in eukaryotes. Eukaryotes have longer linear chromosomes. In the chromosome, at multiple points, DNA replication is initiated by eukaryotes. Replication forks terminate and meet at many points in the chromosome. Though eukaryotes have linear chromosomes, DNA replication will not reach the very end of the chromosomes, but DNA replication ends at the telomere region of the repetitive DNA. The telomere of the daughter DNA strand is shortened. Further, in somatic cells, shortening of the telomere is a normal process. Before the DNA lose 4 bits further partition, cells can divide only a particular number of times. Telomerase can be active in somatic cells mistakenly. This sometimes leads to cancer formation. Termination is the process where the progress of the replication fork must be stopped or blocked. Termination at a specific point involves the interaction between two elements. The two elements are a site sequence in the DNA that is to be terminated. To stop DNA replication, a protein binds to this sequence physically. Termination is the process where a specific segment of the DNA molecule is copied into RNA by the enzyme called RNA polymerase. In the gene expression process, transcription is the first step. During the transcription process, RNA polymerase reads the DNA sequence and produces a complementary antiparallel RNA strand. Transcription includes uracil in all cases where thymine would have occurred in a DNA complement. The steps involved in transcription are: transcription of a gene is initiated by enabling the promoter DNA to RNA polymerase by one or more sigma factors. A transcription bubble is moved by the RNA polymerase over the double helix DNA molecule. It moves like a slider of a zipper. This splits the DNA molecule into two strands. The two strands are broken down by breaking the hydrogen bonds between the complementary DNA nucleotides. Of one DNA strand, matching RNA nucleotides are paired with the complementary DNA nucleotides by RNA polymerase. To form a RNA strand, RNA sugar phosphate backbone is formed with the help of RNA polymerase. Hydrogen bonds of the RNA plus DNA helix break by freeing the newly synthesized RNA strand. In the cell contains a nucleus. Through the nuclear pore complex, the RNA is further processed and gets out into the cytoplasm. At least one gene is encoded during transcription. If a transcribed gene encodes a protein, the result of the transcription will be a messenger RNA. The messenger RNA will be used to create the protein through the process of translation. There are two regions: 5' untranslated region, shortly called as 5'UTR region, and 3' untranslated region called as 3'UTR region. The regulatory sequence, that is the upstream before the coding sequence, is the 5'UTR, and the regulatory sequence, that is the downstream following the coding sequence, is the 3'UTR. The major steps in transcription process are pre-initiation, initiation, promoter clearance, elongation and termination. In eukaryotes, RNA polymerase, a core promoter sequence in the DNA is required for the initiation for the transcription process. In DNA molecule, there are regions called as promoters. These regions promote the transcription process. Core promoters are sequences present in the promoter region that are necessary for the initiation of the transcription. In the process of various transcription factors, the RNA polymerase can bind to the various promoters. A type of core promoter known as a Tata box is a short DNA sequence in eukaryotes. The pre-initiation complex contains core promoter sequence, transcription factors, RNA polymerase and activators and repressors. Transcription in bacteria begins with the binding of RNA polymerase and the promoter in DNA. In eukaryotes, transcription process is more complex. Transcription factors are a collection of proteins. They mediate RNA polymerase binding. This initiates the transcription process. The RNA polymerases will bind to the promoter only when some transcription factors are attached to the promoter. After the first bond production, the promoter must be cleared by the RNA polymerase. During this period, there is a liability to release the RNA transcript. Further, truncated transcripts are produced. This is abortive initiation. This is common for both eukaryotes and prokaryotes. For the RNA synthesis, the template strand is used as a template as the transcription occurs. To create an RNA, copy RNA polymerase cuts through the template strand and use base pairing complementarity with the DNA template. Though RNA polymerase cuts through 3' to 5', the newly formed RNA strand and coding strand can be used as reference points. So, the transcription can be accounted as occurring from 5' to 3'. An RNA molecule from 5' to 3' is produced, which is an exact copy of the coding strand. Two different strategies are used by bacteria for termination. They are row dependent and row independent termination. In row independent termination transcription process, when the newly produced RNA molecule forms a GC hairpin loop, the transcription will be stopped. In row dependent, the interaction between the template and mRNA is destabilized by a protein factor called Rho. The process in which the proteins are created by cellular ribosomes is known as translation. There are 4 phases in translation, namely initiation, elongation, translocation and termination. During the process, the mRNA's nucleotide sequence translated into amino acid sequence of the protein. In the initiation process, an initiation complex is formed. The ribosome binds to a specific initiation site on the mRNA. The initiator tRNA is tempered to the initiator codon and is bounded to the ribosome. In the elongation step, according to the sequence determined by the message, the amino acids are joined to the polypeptide chain. By the same mechanism, incorporation of each amino acid is occurred. The same steps are thus repeated again and again until the termination codon reaches in the message. The third and the last stage of the protein production are given by the termination codon. Termination occurs when the ready-made protein is released from the ribosome. The translation process also requires a large number of protein factors that helps in the binding of mRNA and tRNA to the ribosome. A large part of the energy produced in a cell is consumed by the protein synthesis. Hence, in this session, we discussed in detail about the central principle of life. We also discussed the process of replication, transcription and translation. The genetic information passed through replication, transcription, translation is shown by the central dogma. DNA is transformed into RNA and again RNA is transformed into protein. The process of developing two same exact or identical copies of DNA molecule from one original DNA molecule is known as DNA replication. Transcription is the process where a specific segment of the DNA molecule is copied into RNA by the enzyme called RNA polymerase. The process in which the proteins are created by cellular ribosomes is known as translation. The genetic information involves in the development and functioning of the human systems.